So welcome back. Hopefully everyone had a good quick lunch break. This morning we heard a lot about some newer methods for mapping M6A in mRNA and quantifying M6A in mRNA. This afternoon, we're going to pick up in a sense where we left off and focusing on mRNA modifications, a lot on M6A, but talk about some newer instrumentation for detecting and quantifying these RNA mods, and with a particular focus on nanopore sequencing. So we have our first speaker for this next session is going to be Dr. Jonathan Goki from the Genome Institute of Singapore. And the title of his talk is Identification of M6A RNA Modifications at Single Molecule Resolution Using Nanopore Direct RNA-seq Data.
All right, yeah, thank you very much. So thanks a lot also for the invitation to speak here. So I'm based in Singapore. It's a bit early in the morning. I might not be able to join the panel discussion, but any question, there should be time after my talk, and also if you post it to the chat, I'll try to reply to all of those. Today I will be talking about nanopore sequencing and detection of M6A. We're actually a computational team, so we're interested in transcriptomics data. Here you can see an illustration. This is one gene that is transcribed into RNA. On this side, you can see all these RNA copies. And one thing that fascinates us is that these RNA copies can be so different from the gene. It's because there's different exon combinations, steroid and end sites. And of course, the topic for today, the RNA modifications. They don't need much of an introduction. We have heard a lot about those already. Very briefly, RNA modifications are chemical alterations of nucleotides, and they're regulated by RNA-modifying enzymes. And there are actually quite a few of them. This just shows an illustration of mRNA with some of the modifications that are there. And I will be talking about M6A today, which is the most frequent mRNA modification. Again, it's really nice that I don't have to talk much about the motivation for doing this here, but very briefly, it has been involved in a lot of different aspects of RNA biology, and we have heard more details than what I have here on my slides already. And it's also impacted in diseases with some of the most striking examples coming from cancer. So it's important that we find out where M6A is located. And one way this could be done is, in a way, one way or another, what we try to do is we try to identify the RNAs that have this modification, what you can see here on the right. And if we use short-read cDNA sequencing, we try to identify those parts of the RNA, map that back to the genome, and then we can identify those positions that have a modification. And that's what we call a site-level modification probability. Okay. So what we learn from that is we can find out which genes generate these modified RNAs. But what we actually want to know is more the picture here on the right. We want to know which RNAs are modified. Well, that's slightly different, but if we find out which RNAs are modified, then we can ask questions like how many RNAs are modified? Do these RNA modifications co-occur? And are they associated with RNA processing? The technology that I'm working on is nanopore sequencing. And so to very briefly introduce that, the technology works by pulling a nucleic acid through a nanopore, and that can be DNA or RNA. And each time point, an electric current signal is measured. And so the signal looks like this. From that signal, we can then identify the nucleic acid sequence. And the interesting part, of course, is that we can sequence the native DNA or RNA molecules. And that means we can identify potentially RNA modifications from that signal data. To illustrate that here for the central position, imagine a methylated A sequence that appears as somewhat a shift in the signal data. And that's what we can use to computationally identify those modifications. And this kind of scenario here where we have two different cases like modified, unmodified, or red and blue, that's perfect for supervised classification or binary classification. The only issue is it doesn't exactly look like that. Okay? It looks more like this here. We don't have the colors, and that's because we don't know which reads are modified. The only thing that we know is that at this position, there's a modification. So that brings me back to what I've shown previously. Now we have the data for individual RNAs, for individual reads, but the label that we can use for training any supervised method is still at the site level. And so we still have this mismatch here. This problem is actually known in the machine learning literature as a multiple instance learning problem. So to briefly introduce that, I use this example here. Imagine there's 3 security guards, and all of them have their keychain, but only 2 of them can open a building. And the reason that they can open the building is, of course, that they have the right keys. And so here, the keychain, that's what we call the bag. The keys, those are the instances. And that's the multiple instance learning problem, where we have the label only for the bag, for the keychain or for the security guard, but not for the individual instances, the keys. And that's the same picture if we look into RNA. What we have here is this kind of bag that's the set of all RNAs, and the instances are the individual RNAs. And the label that we have is that we know that at this position, the RNAs might be modified, but we do not know which of these RNAs are modified. And so what we have done is we have implemented this method that's called M6ANet that uses the multiple instance learning framework to infer the M6A probabilities. And we can do that from a single run of direct RNA sequencing data. So this very briefly works by using the signal data and the sequence data, which is really informative as well. In this case, it's mapped into a high dimensional space to this read level feature encoder. And then in a second encoder, we'll calculate read level probability. And then the third step is that we use those to infer the site level probability. So the beautiful thing about this is that everything here is learned end to end. So the only thing that we need are those site level labels. That's what we can get from other experimental data. And the read level features and the read level probabilities are all learned end to end implicitly. So to show how that works, just with one example first, let's look into the site level probability for this example gene here with in green, the M6A-seq check showing M6A sites. And in blue, you can see how this looks for the predictions that we get from M6ANet from direct RNA sequencing data. And it looks pretty similar. The nice thing, of course, is that we also get the long-read RNA-seq data. So we get the transcriptomics information as well. And it's just a relatively simple assay, which means we can also easily scale it. And that means we can look into a lot of different cell lines and start comparing them. Now, this is just one example. How well does that work if we look transcriptome-wide? A classification method is usually evaluated with a plot like this. That's called a ROC curve. Shows the true positive rate over the false positive rate. And so the higher the curve, the better. And what you can see here, that M6ANet in blue shows a very good performance compared to other methods. For completeness, this is on all M6A k-mers. We also show this on subsets because not all methods work on all, but the picture looks very much the same with M6ANet, showing a very good performance. Again, for completeness, there's a second plot that I'd like to show that's very commonly used, in particular for what we call imbalance data. And that's data where we have many more, in our case, unmodified sites compared to modified sites. It's a precision recall curve. The main thing, again, is, in comparison to the other methods, M6ANet shows a higher precision at almost any point of recall. And there's a lot more information in these plots. I won't talk about that now, but if anyone has questions, I'm really happy to discuss more about that. Okay, now what I've shown is how well M6ANet works if we train it on a human cell line. We keep some genes apart that we don't use for training, and then we test it on those genes, and that works really well. But what we want to have is a method that anyone can use on their own sample, on their own cell lines that they're working with. And so we want that M6ANet can generalize very well. And so the way we wanted to evaluate it is by training it on a cell line like a kidney cell line, my HEC293T here. And then we wanted to apply it on the colon cancer cell line, and then that's what we did, and it works really well. And then we thought, okay, it seems to work pretty good. And we had excellent reviews, I have to say here. And there was a suggestion from 2 reviewers who said, well, you should look into data that's a bit more different than that. Why don't you look into a species that has a very different k-mer context for M6A? And so the suggestion was to look into Arabidopsis. So the question is here, can you train a model on Arabidopsis and still identify M6A in a human sample? And so that's what we did, and it works really well. It's what you can see here in green. If we train on Arabidopsis and we make predictions in a human cell line, the accuracy is still very high. And the gap that we can see here, we probably know where it comes from. It's not necessarily that the model is not robust against this variation in species. It's probably more that the way that the training labels are defined, which is slightly different. And in any case, the gap here between the human and the Arabidopsis model is smaller than the difference between any of the other existing methods. So overall, I think this really shows that M6ANet is very robust. It means you can really apply it on your sample. I hope I've convinced you, but there's also other people who have looked into that. And there was this recent benchmark that compared supervised M6A methods. And again, they used a different data set than we used. That's, I think, mouse. But again, M6ANet really shows a very good performance here. Okay, so this all shows that M6ANet works well on Oxford Nanopore sequencing data. How does it compare to other technologies? And so if we compare technologies for detecting M6A, it generally looks something like this, with good overlap between technologies. And that's where we often see the highest confidence size, those that have a lot of data supporting them and that have very high modification rates. But all of these technologies really have this large number of sites that are very specific to each technology. And it doesn't mean that they are wrong. It just shows really how different technologies are. And one thing that you can already see here, it's not that nanopore sequencing is different to everything else, just that all the technologies differ. And so we try to look into that a bit in more detail. One way to look at that is we're looking at whether these sites that we identify are sensitive to metal 3 knockout. That means we'd expect that the M6A sites are lost. And for all of these sites, we do see that that they're sensitive. M6ANet here shows a bit of a better signal, but it might be biased towards nanopore sequencing, which we're using to evaluate that. Another way to look into this is to see if the enrichment is as expected. We've seen those plots before. We want to see that the M6A sites are close to the 3' UTR. And again, that's what we see for all these methods, even if we look into those sites that are unique. So I guess the summary from this is really not very conclusive in saying that one of the technologies is better, but I think one thing that one can see is that nanopore sequencing is really a very promising technology. And there's still a lot of other aspects that I think makes this a very promising technology for looking into RNA modifications. One is actually an aspect that I have mentioned in the beginning and that I will talk about in the next slides. That is that we can look into those single molecule M6A predictions. Just to illustrate that, the first thing I want to show is this kind of read level feature encoder that maps those read into a high dimensional space. And that space by itself is not informative. It's just optimized for the classification task. But you can use that and reduce it to 2 dimensions. Okay? So it really helps us to just identify those or just visualize those reads. And that's what you can see here for one site using a sample, a wild type sample, and a metal 3 knockout sample where there's loss of M6A. You can see that those reads then, here each circle is showing us an individual read, that they separate separate clusters separately. We can also do that with synthetic sequences. We can annotate these clusters and see that they really map into a modified and unmodified cluster. This is purely for visualization. What we can do is we can also calculate the probability that a read has an M6A modification at a very specific base. So how well does that work? So to evaluate that, we used synthetic data, and we mixed synthetic data, which has 100% M6A modifications, with synthetic data that has no M6A modifications at certain ratios. And then we try to predict that ratio using these single molecule probabilities. And what you can see here is that it's very close to what we would expect. So that's as good as it gets. I want to say that we use this data also to calibrate the probability, read level probability. So it's optimized for that task. We can also look into that using human cell lines. What we have been doing here is use samples where we mix RNA from a metal 3 knockout cell line with the wild type cell line at specific ratios. And so the relative modification rate is, the average relative modification rate is what we know. And then if we compare that with predictions from M6ANet, you again can see that it aligns quite closely. So overall, I think this really indicates that the read level probabilities that we identify seem to be quite accurate. Okay, to summarize the first part, I've shown that M6ANet seems to accurately identify M6A from direct RNA sequencing data. It has a very good performance also compared to other methods. And the single molecule predictions seem to reflect the expected modification rates. I've also shown that M6ANet generalizes to new cell lines and species, and it was one of the key aspects when we designed it, because we want to make sure that you can apply it actually to any sample of interest, not just to the one that we have used for training. And so that's really clear. You can do that. There's no retraining and no additional data required. You can just apply it really out of the box, at least for species that are similar enough to Arabidopsis and human, that is. There's one more exception, and that is, what if there's a change in nanopore sequencing chemistry? Now that will change things more than the species. And so we recently tried that out. And so many thanks to Oxford Nanopore for providing early access to the next iteration of the direct RNA sequencing kit, which is called RNA-004. Everything that I've talked about before was RNA-002. That's what's currently available. And RNA-004, really, first thing we see is that there's a very good improvement in throughput. With one run, we get more than 17 million reads. And at this point, this is so much data that we almost think it's too much data. So I think that's really very nice to see. The other thing is that the signal data that we're working with is very different, and that breaks everything really. That means we cannot identify M6A just using that model. We're very lucky that we had really great help from Hasintu and his colleagues at the Garvin Institute, and they solved the first issue that we had, and that was that we couldn't process the data properly. And so now that's possible. Steps called event alliance, and so we can process the data. And then the second is that we retrain M6A-Net using data from RNA-004. And so just to show how this looks, I had many thanks here to say to Christopher and UK as well. This is how the comparison looks between the model of 002 and 004. This is on the intersection of sites that are detected by both, but what you can see is that really the performance is, I would comparable. The 004 is not evaluated in as much detail as the 002 model, but overall it looks really good. Another note, of course, is that because there's so much more data, you get many more sites with a new kit, and that means it's much more sensitive. But of course, there will be a lot of sites that might not be in the set of labels that we are using. Okay, in the last minute, I just want to briefly mention that there's another way to look into RNA modifications from direct RNA sequencing data. That goes back to that picture where you have the blue and the red, and I said we actually don't have those labels for individual reads. But in some sense, we can use that information, and that is if we have samples that we want to compare. And so our colors would be those samples. And that's a slightly different design. Now we want to look into differential RNA modifications. So this is one of the most important tasks actually in transcriptomics data. And so I like to show this, but this paper is actually really nice. I'm taking this out of context here, but just as an illustration of differential expression analysis. One of the most commonly used tasks, we want to identify how much of a change do we observe, the effect size, and how significant is it. And so the same thing, that's what we can do also with direct RNA sequencing. Can estimate the effect size, which is what you call the differential modification rate, and then the significance of that observation. And so the method that we have developed to do that, that's called XPOR. So it uses a 2 Gaussian mixture model to identify those differential RNA modifications. And it really can be applied in a case where one has this kind of case control design. It works for M6A modifications. That's where we have evaluated it. But in principle, it works on any modification change. And just to illustrate how that looks on real data, this is a comparison of the wild type cell line with the metal-3 knockout cell line. And each point here in those scatter plots is an individual site. And you can see those sites that do not show any change are on the diagonal. And those sites that are lost after metal-3 knockout, and those are shifted. And those dark blue points indicate that those are significant shifts. And you can apply that, of course, if you have a matched control where there's a loss of a modification. But you can also apply it to compare other cell lines. It doesn't have to be perfectly matched. For example, you can compare another cell line against the HEC293T control. That works well. We can even apply it on clinical samples. And that really shows that this is actually a pretty flexible framework in terms of direct RNA sequencing. That's also something that's possible on clinical samples as well. And so I think that's also a nice way to show that this approach might sometimes be helpful if one has a good control to use. This was also actually part of the benchmark paper. And again, I think it was nice to see that XPOR actually shows a very good performance. Okay, so this is actually my last slide that I want to mention. If any one of you is interested in direct RNA sequencing or long read sequencing in general, but has not worked with any such data or has not generated any such data, we have made this huge data resource publicly available. That's called the SGNext project. It contains sequencing data from multiple cell lines and with a lot of different RNA sequencing protocols, including direct RNA sequencing. There's also M6A-seq data available. It's all hosted for free on the AWS Open Data Registry. And that means you can very quickly access it, which is particularly helpful for direct RNA sequencing data, as these are very large, these files. And so there's also online tutorials that you can look at. So if you're interested in that data, I really encourage you to have a look. And with that, I'm coming to the summary. I've talked about nanopore sequencing for detection of RNA modifications, in particular M6A, and I've introduced M6ANet, which uses this multiple instance learning framework. And it learns model parameters using the site level labels, but implicitly you can also infer the read level probability. It shows very good performance and it generalizes very well. And it estimates single molecule M6A predictions that seem to reflect the expected modification rates. I very briefly mentioned also that you can analyze differential RNA modifications if you want to compare samples, and that can be done with Explorer. And again, I just want to highlight this data resource, which is publicly available and can be used if you're interested in that. That I'd like to thank everyone involved, and that's particularly Christopher Hendler, who developed M6ANet, and also Yuki Wan, who has helped in all our RNA modification projects and implemented the RNA-004 model. Janine, who has worked on the SGNext data, as well as Floyd Patawanish, who has developed Explorer. And with that, I'm happy to take questions.
So I think we have time for a few questions here, and we have a few in the chat. So Logan Maroney was wondering if you compared your M6ANet sites to Glory at all, or for that matter, maybe ETAM-seq.
Yeah, no, we haven't done that. Yeah, I'm interested in that. The simple answer is we have not done that, but I think it's something I wanted to. Yeah.
And I'm actually curious, and Hung Mai posted a question, I think, right before you talked about your differential methylation analysis. But I'm curious what the sensitivity is of differential methylation that you can detect.
We actually have done a relatively detailed analysis of that using these RNA mixtures. So what we did is we took the HEC293 cell line, the wild type cell line, and the METO3 knockout cell line. And we mixed it at different ratios. So we mixed the RNA at 25%, 50%, and 75%. And then we asked how well can we detect differential modifications. And with 50 and 75%, it works very well. With 25%, it still works, but we do see a more significant drop. We also looked into what happens if we don't use a knockout cell line, but a knockdown, which is a partial knockout in a way. And that's also something we can detect, but we already see this kind of shift. So it works much better if there's a very strong signal. If the signal becomes weaker, then it gets more difficult. And one thing that I think will make a difference with more throughput, I think it ends up being much more sensitive again. So yeah, we see also one can use replicates. And if the throughput is not very high, sometimes it makes sense to pool the replicates together. Rather than treating them separately, then you get more coverage for more reads, for more positions. So that also means there's two ways in terms of sensitivity. One is with low stoichiometry. The other one is global transcript coverage, and pooling data can help there. But yeah, with the PromethION and the new RNA-Kit, I think throughput isn't an issue anymore.
Yeah. And maybe at least one more question here. Kandarp Joshi asked a few questions, one of which is, what is the optimal depth of nanopore direct RNA-seq and RNA-002 to capture? Say, maybe you can compare to the more recent, version as well.
Yeah. So with direct RNA sequencing, there's no multiplexing, and that means there's only two options. One is the GridION or MinION device, and that gives you about 1 million reads. The other one is the PromethION device that gives you maybe 4 to 5 million reads, sometimes a bit more on the RNA-002. Overall, I think the GridION runs are sufficient. One can do a lot. 1 million reads means really a lot of data to work with. One wants to have more sensitivity, and the PromethION runs are good, but with 5 million reads, I think that's more than enough. as I mentioned before, with a new development kit that goes into a range everything is too much. So I think a multiplexing option would really be something that could make a huge difference. Then we can start putting samples together. And so then the question becomes even more important, because then you can optimize the cost. Currently, it's really just two options. There's no multiplexing. And I think sometimes it might be more helpful to have more replicates and then putting the data with the GridION, rather than having a lot of Promethean runs, which gives you a lot of data. So at this point, in terms of also of cost, multiple replicates on the GridION might be good.
Yeah. Well, I think in the interest of time, we'll have to stop there, but thank you for staying up, and we hope we can see you in the panel, because I think some of these questions are important to further talk about. But if not, hopefully at least you can address some of these unanswered questions in the chat. Thanks everyone. These are great questions. Sorry we couldn't get to all of them, but thank you very much.
Thank you.
And up next, we have Dr. Shuo Hong from Nanjing University, who's going to talk about identification of epigenetically modified RNA nucleotides using a reactive nanopore sensor.
All right. So hello everyone. First of all, I'm really glad to be invited to give this talk. I really apologize if you find I'm not in a very good state, because I'm recently very sick. But I still will do my best to finish the talk. So today I'm going to talk about like a new technology that is gonna recognize epigenetic modified RNA nucleotides. I apologize that this technology will not, well, at least at this present form, it will not do sequencing. It's more fundamental, and but in the near future, we hope it could help to optimize how nanopore sequencing could be done using this kind of technique. So a very brief introduction of myself. I graduated from Nanjing, and then I work in Nanjing as well. But before that, I was working with Professor Hagan Bailey as a postdoc in University of Oxford, where I learned this kind of nanopore technologies. So this is today's outline. I'll give you like a very brief introduction about what nanopore is. I guess everyone knows about it, but from the fundamental aspect of view, it's like a channel. And actually, I like protein nanopore is because it's very easy to make and to engineer, even in a small size lab like MySense. And then my focus, although there are so many different nanopores, my focus is more about the MSP nanopore, and I will talk about why I focus on that. So solely with MSP nanopore, we actually can do like a wide range of analyte sensing from like a single proton or single metal ions to a whole piece of tRNA molecules. So it has a very, very large dynamic range. But actually, I guess people in this conference is more interested about like RNA. So nanopore can sequence RNA and DNA in the way that everyone knows. The only thing that is making me not satisfied is because like this is a piece of DNA with just a simple sequence, but its generated nanopore sequencing data is very complicated. So that's why we need like bioinformatics and algorithms to decode them, which I feel is very complicated. And this is the RNA sequencing version, and still is very complicated. So in my opinion, as a chemist or nanopore chemist, I hope we can invent a technology that I can just give you read of A, U, C, and G pretty much directly, and no other like no other combinations of them. So that's why we need to introduce a new technology that most people actually doesn't know about it. So it's just called a nanopore single molecule chemistry. It's a kind of like a concept that Professor Hagan Bailey started in 1997. So it's a pretty old concept, but still not very many people know about it. So we know that a lot of protein actually combined with metal to have some new functions. For biological nanopore, you can engineer that to have a metal binding site. So in this way, you will have like a reversible chemical reaction happening in the lumen of this nanopore. So this reaction, though it's very subtle, but actually could be monitored by this biological nanopore in a very precise way. So we call it nanopore single molecule chemistry, but in another word, you can also call it a reactive nanopore because you are using this reaction to monitor a very complicated or minor different chemical structures. So to me, I never intended to invent a technology to say immediately sequence RNA or DNA, but actually by any accident, we noticed that with an MSP nanopore, someday we realized that it can also monitor chemical reactions. For example, this is a tetrachlorate. It's like a single onion. When you put that into this nanopore, some magic will happen. Basically, with this sulfur-containing amino acid right at this position of the MSP nanopore, it will react with tetrachlorate in a highly reversible way, and then you see this kind of reactions. And they actually correspond to this intermediate state, which will last like just a few milliseconds and then disappear, go back to here. And at some point, it will go to the end point, and it will never go back to this kind of reversible reaction. So basically, using this nanopore, you are monitoring like a very detailed chemical reaction process. So why I'm talking about that actually is because this is the first discovery of this nanopore properties because it has an exceptional atomic resolution. So these event amplitudes are extremely large. It's because it has a wide opening which focus on this spot so that it's actually amplifying your event amplitude. So it has very good resolution. So the next thing is saccharide. You might be surprised why I'm talking about saccharide. It's because actually saccharide is a next step towards RNA. So these are 4 saccharides, monosaccharides. They have exactly the same molecular weight. So with this kind of nanopore, we still have another thing to do, which is to make it heterooctameric. So this MSP nanopore is octamer. So that actually means engineering of this nanopore is very difficult. But instead, we can actually make it heterooctamer so that it will look like that. So one of the monomer will be different from the others. And this monomer will contain a unique reactive site so that it can bind with, for example, a phenolboronic acid reactor. And then this nanopore will be used pretty much like all other nanopores, but it will start recognizing different saccharides. So it's because of this these pairs of hydroxy groups which reacts with the phenolboronic acid. So even different saccharides with the same identical molecular weight will be differentiated using this nanopore. So actually, with this kind of demonstration, we know it has extremely high resolution, even if you analyze about the same. So with this kind of resolution, we have successfully discriminated almost all monosaccharides that we can have from the market. And they are discriminated, and using machine learning, we can evaluate its performance. It's about like 97% accuracy. And the reason for this remaining 2% something is because even the monosaccharide itself is not 100% pure. So we have included this kind of impurities into our machine learning algorithms. So that's why we can have this kind of errors. But actually, it's very, very high resolution. And then taking this step further, we have approved that we have very high resolution. So why not just give it a try to something more challenging and also more urgently needed in the academia. So then some of you might have read this kind of paper from Nature Nanotechnology. We were fortunate to be selected as the journal cover. And also this some reports about nanopore distinguished RNA modifications and so on. So the technical principle is almost identical to that we used with the monosaccharides. So basically, we build an MSP heterooctamer. It's not easy, but actually that's definitely worth the effort because afterwards you can have a single soil group at the pore constriction. It will be modified with this phenolboronic acid, and so that you will have this phenolboronic acid here inside the pore lumen. And this is a nucleoside monophosphate. We call it NMP. The NMP contains this kind of C-style structure because this part is a ribose. So it will reversibly react with the phenolboronic acid to form this kind of structure. Because different RNA nucleotide will have a different base. So different NMPs will be discriminated using the resolution provided by this MSP nanopore. So this is the first data, like a mixture set of data using like nucleoside monophosphate composed of A, U, C, and G. Like I expected, I actually can see only C, G, A, and U, this kind of readout. So these are my data readout. And I can confirm that these are not computer generated data. This is the raw data. It's just it's an either or relationship because can't be anything in the middle. So that's why it's very high resolution and it's very clean. And again, like when we submit paper, the referees also ask us like to verify this technique is definitely reading the differences between C, U, A, and G. So with the same pore, we can sequentially add like NMPs of C, and then further add U, you will get like a second peak, and then add A, you will get third and fourth peak. So basically, we can confirm that it's definitely from the AUCG. It's not from like systematic error from different pores or so on. So this part is confirmed. And with this kind of high resolution, so what else we can do? We know, actually, I learned a lot from Chen Qi from Peking University that the RNA is heavily epigenetic modified. So, and there are so many different modifications. But we, from the market, we can only get like 14 NMPs as a standard. So they are basically pure chemical compounds so that you can test base. And then these are pseudorudin and M5C and so on. So they are only slightly different. And it's very interesting that this M6A and M1A is pretty much the same molecular weight as well. And there are also many others, but we just can't get their standard compound to test base. So we just tested 14 of them. And then just with minutes, they will be discriminated using this kind of technology. So here, actually, I can show you the raw data of this demonstration. Well, it looks like nanopore sequencing already, but actually it's not. It's just a separate RNA nucleotides. And whenever it's modified or not, we can immediately tell. And this is pretty much the actual playback speed of our data acquisition. So you can actually see it's very clear. Even with your naked eye, you can tell the difference. And also, we also tried to compare using the same nanopore to see the difference between canonical and epigenetic modified version of the NMPs. For example, C versus M5C and G versus M7G and so on. So you can actually see that they are very easily discriminated using the high resolution dopol. So when we submit paper, we were very excited and we thought the technique is pretty much ready to approve its resolution. And then the referee gave us like a even further example to do. So the referee actually asked us to do like a natural piece of RNA containing heavy epigenetic modifications. So the first thing that we can think about is a piece of tRNA. So this is a yeast tRNA sample. According to its structure or sequence, we know that it has a lot of epigenetic modifications on it. We realize that some of them has already been done by us. For example, this M5C and M7G. And some of them could be detected like M2G and M22G. And some of them could never be detected. I will talk about why. So we just enzymatically decomposed this whole piece of tRNA into nucleoside monophosphates and put the whole mixture into our nanopore sensor. And then we will get like a very complete spectrum of its RNA modifications. So these color-coded clusters are actually just like standard data that we already have in record. It's a perfect match with the tRNA data. And there are some other like unidentified clusters. For example, 1, 2, 3, and 4. So we have 4 unidentified clusters. They actually correspond to this M2G, M22G, Y, and T. Because we don't have the standard sample of them, so we call them unidentifiable, but they are definitely detectable. So we just don't know which one is which, but they are definitely detected. And for other epigenetic modifications like D and M7G, and it's very surprising that M7G actually give a very unique signal. It's very large in amplitude. And then we quantify them. it's very consistent with the RNA sequence of this tRNA sample. This is the true value and the measured value. They are very consistent. So basically, even we can't sequence the RNA, but we can actually quantify the RNA modification of this piece of standard sample in a very accurate manner. So here are the pros and cons. Well, I think the biggest cons is still not yet ready to do RNA sequencing, but the pros is that it has a very high resolution. Here, there are some samples. We know they are detectable, but we don't know which one is which because we don't have the standard to test with. But whenever we have the standard, well, just to put that into the machine learning algorithm, they will be automatically identified. And there are also some other things that we apologize for because this part has this methoxy groups instead of hydroxy group. So it will never react with the PBA reactor, so it will never be detected. So we apologize for that. But these part are a majority of the RNA nucleoside monophosphate. So most of them could be detected. So, and also, yeah, we know like nucleotides could be in different phosphorylated forms like NMPs, NDPs, and NTPs. And even they are combined with epigenetic modifications, they will also be detected simultaneously by this kind of same nanopore. So here is an example of all 12 combination of them. So with this simultaneous presentation, you can actually see how well they are discriminated as well. So to summarize, so I'm actually demonstrating a heterooctameric MSPA, which we recently invented. So it's very useful because this heterooctamer allows for an accurate and precise engineering of this pore by having a chemically reactive adapter. So this pore in the hull becomes reactive to specific nucleotides. And the PBA actually allows for this kind of reaction, and the high resolution actually comes from this conical pore lumen. So I don't know whether you know that actually MSPA was the first nanopore that successfully achieved the nanopore sequencing of DNA. It was not the CSGG, which is later. The MSPA actually have a very high resolution because it's this kind of conical shape. So although I can't sequence RNA using this pore at this moment, but future development may enable that. And also it's very good to discover like new RNA modifications. For example, if you go back to this kind of event cluster, like a plot. So if your RNA sample by any chance having like a new kind of modification that nobody has ever discovered, it will definitely appear as a new cluster here. So it's very useful to discover new like RNA modifications as well. So eventually, I would like to acknowledge my founding bodies and also my great students who performed this work. A majority of the work was performed by Yu Qing, who is an independent PI in Nanjing University already. So that's all of my presentation. Thank you.
Absolutely. So I have one question real quick. So,
sure.
You know, obviously this has a lot of promise, and as you just said, there are a lot of advantages to using, you know, this nanopore. How far off do you think we are from being able to use this to do, you know, transcriptome-wide sequencing, and what are the major barriers in your mind that need to be overcome to get there?
Okay, so I think the biggest difficulty is to retain the sequence order of these NMPs from their original sequence. And we try to use like enzymes and conjugate that onto the top of the pore. So it could work like a trap-and sequence mode, but it's not as easy as we thought. So we successfully did this kind of pore conjugation. It's all right. We had a lot of troubles to make the pore active and the enzyme active at the same time. But even we can do that, whenever this like this RNA nucleotide has been chopped off, we can't guarantee that it will immediately get to the pore and also react with the PBA. So there's a big chance that it will just escape from the RNA, but escaping to the buffer solution and never be captured anymore. So this is the most big difficulty at this moment.
Makes sense.
Yeah.
Let's see, we'll go to the chat, see if we have any there. So Veronica De Jesus asks, if you can detect RNA cap structures. I mean, I know you showed with the NMPs things like M7G, which are certainly part of the cap. This question might refer to more extended cap structures.
Yeah. So as long as the C style still retained, we can always detect them.
As far as the
as long as the C style structure of the ribose is retained, we can always detect them.
Yeah, that's great.
Yeah. And also, so far we never seen any two NMPs that can never be distinguished. We have tested about like 14 of them, and we have never seen two that is 100% overlapping. So they'll always be discriminated. Yeah.
Yeah. I mean, it seems like, with the data you showed, you're getting such good resolution. It's just incredible, even with highly similar, chemically modified.
Yeah, that's right. We are also surprised too, like the M1A and M6A.
Right, that's what I was thinking of, right.
Yeah, it's very cool.
Yeah, I mean, especially, coming on the heels of the last talk where we saw that there are, great methods like M6A-NET, but then, Jonathan also talked about there are many other methods. And I think those who might have, be aware of this part of the field have certainly read the papers of all these different methods that claim to be the next best analysis, method for nanopore M6A data. I think it's definitely very promising if we can get something that has, this type of, clean signature already. But as you mentioned, we have a ways to go before we can
right.
be able to implement
this.
Trying that.
Yeah, definitely. And another thing to achieve this high resolution is because we chopped the RNA into pieces. So the limited spatial resolution of the pore has been like minimized, because anyway, you're just recognizing like a single RNA nucleotide instead like a whole piece. So if you sequence them in a whole piece, anyway, you'll be entangled from other neighboring RNAs. But in this way, they will be separated. So that's why you have like a better resolution.
Mm-hmm. Yeah. I think we have time for one last question here.
Yeah.
Ancon Banerjee asks, can cyclic NMPs be distinguished from NMPs?
Sorry, where is the question?
And can cyclic NMPs be distinguished from NMPs?
I don't think so, because the cyclic NMPs doesn't have the C style, right? If I remember correct. Yeah, so it has to have a C style structure first. Otherwise, we'll just never react with. And also the reaction, it has a very big advantage because the reaction actually gives you like a millisecond or 10 milliseconds time, so that it can have a good data acquisition. That is why we need the reaction. Yeah.
Yep, that makes sense to me.
Yeah.
Okay, one last question from Puneet from Famacetti. A thought that, thinking of if you can use specific reactivity of certain modifications that are known, if you can kind of use that knowledge to identify unknown modifications.
Yeah, I think so. So actually, that's the one of the few advantages of this technique, because so far we haven't seen any two NMPs that is having like overlap nanopore features. So if you have like a very complicated, new, never discovered NMPs that is epigenetically modified, and put that into the nanopore, you will very likely will report like a strange signal. According to the machine learning algorithm, it will just give you a feedback. So this guy I've never seen. So you are repeating this, seeing this kind of new nanopore events, and they will appear as a cluster in the event scatter plot. So then okay, I don't know what that is, but there are definitely a new guy in this RNA sample. So it can give you some kind of feedback that something new is happening. And then you might take your sample and put that into like a mass spec and see what that might possibly is. And then you can combine like many different techniques to confirm what it is. So, but the first thing is you can use nanopore to detect this new modification.
Right, exactly. And I think, like you said, the validation is going to be critical.
Yeah, definitely.
Be a nice first pass to
exactly. And also, it's very convenient. Whenever you decompose the RNA into pieces, you just need like maybe 10 minutes of data acquisition. You can get like thousands of data. and that will help you to judge whether you have any new modification or not.
Right, right.
And also, I know that some are recent reports that some RNAs could be like glycosylated.
Yep.
And the saccharides, the glycans on the RNA can also react with the PBA. So that actually means you might, for the same kind of glycosylated RNAs, you might have two set of signals. One is the glycan part and one is the ribose part. So then from that kind of nanopore feature, you can say, okay, this guy must be, well, it's either two kind of new signals, or it could be like same thing that's reporting two signals. So yeah, so that will give you some clue.
Right, right. Well, very exciting stuff. Well,
Yeah.
thanks again. And I think we'll move on to our next speaker.
Okay.
So next we have Eva Maria Novoa from the Center of Genomic Regulation, who's going to be telling us about decoding the epitranscriptome at single nucleotide and single molecule resolution using nanopore sequencing.
Okay. Thank you very much. Are you seeing now the full mode, I guess?
Yep, we're good.
Okay, awesome. So thank you very much for having me here. It's a great pleasure to present a bit of what we're doing in this angle. So following the previous chats, I will also talk a bit about nanopore sequencing. Let me just move this. Sorry, I'm trying to move my screen so I can see everything. Okay, now, sorry. Yes, so basically, also just to briefly introduce our lab, we are the Epitranscriptomic and RNA Dynamics lab. It doesn't need an introduction to this audience, but very briefly, we're interested in modifications, and we do a lot of technology development to study them. A lot of what today I will be talking about is nanopore sequencing. But in addition to method development per se, we're actually interested in applying these methods to answer different questions. Some of them are at the molecular level. So what is the role of modifications in the RNA fate? And also some of them are related to diseases, why their dysregulation is associated to different human diseases, and even across generations, like how, what is the role of modifications even in intergenerational inheritance? But today my talk will be purely methodological and focus on our efforts in trying to detect modifications using nanopore sequencing. So as the previous talk have briefly mentioned already, nanopore sequencing offers the possibility to sequence individual RNA molecules, including their modifications in their native state. So we can capture virtually in principle any given modification, and including from M6A, which has been the focus of previous talks, and also like what I will be talking about, but also as it has been shown previously, it can also be used to detect other modification types. So I don't need to explain a lot about the advantages that it has, but I would say that what is important also is to keep in mind the challenges of this technology. So first of all, is like we always say, yeah, we can detect modifications, but the truth is that there's no base caller for modifications. Another limitation is actually the large amount of input that it requires. Now, these values are decreasing, and like the latest kit version that the RNA-004 is supposed to work with decreased amounts of poly-A selected RNA, but it's still a huge amount of poly-A selected RNA, which makes it a bit challenging for certain applications or patient-derived samples. The standard protocols are limited to poly-A plus RNA, and also the standard protocols are limited to long RNAs. So there's all these challenges which really limit the applications. And today I will basically discuss our approaches to tackle these two different challenges. So I would like to start about how to detect modifications. And I would say that in general, there's like three main ways that one can think about detecting modifications. The first of them is using differential base calling errors. This has been developed by some software that we have been doing, but also by other teams as well. The idea here is that when you have a modification, there is a signature that kind of looks like this here with the asterisk of a base calling error, because the algorithm doesn't really know what that modification is. So it sometimes puts somewhere between, in this case, an A, a T, and a C. But then the proof that this is not just a random base calling error, but really a modification, is that when you have a knockout, this error is actually gone. Another way to approach the detection of modification that has also been exploited by many different softwares is the use of alterations in current intensity. So basically, wherever you have here the position of the modified side, sometimes it can be a bit more upstream or downstream. And also it can affect several positions. You will have a shift in the current intensity. Also, it can be associated to a shift in the dwell time or other features, but basically the shift in this current intensity, it can be used to identify whether a position is modified or unmodified. However, both of these approaches, which are basically how we englobe pretty much what is developed for RNA modifications, have several big issues. The first of them is that you do per site modification of prediction. So even if you use per read features, the final prediction is typically per site, limiting our detection to modifications to things that are, like at least 10, 20% modification stoichiometry levels. And also it requires a lot of post-processing, which is a lot of computational effort, and also a lot of biases as well, right? Because by doing the sequencing, base calling, mapping, resquiggling, which this point here, I think it has a major influence in how we bias which reads are actually resquiggled or not. So all these post-processing also biases which reads we can detect or not. So there's actually a third way to detect modifications, which would be the use of modification-aware base calling models. So the advantages here are that one would be in principle able to predict modifications during the sequencing run. And this would really allow you to predict modifications at per read and per nucleotide resolution. So as shown here, right? So what one would expect is that here, for example, each individual line is a read. And here it would be colored by the modification probability. So here, for example, you have an M6A, 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 and M6A. And here in the knockout, most of it should be gone, right? However, unfortunately, this is only available for DNA modifications, and that has been released by Nanopore, but then there's none for RNA. So what would we need to actually do this? Well, we would need to develop a modification-aware model. So instead of a model that predicts ACGU, a model that predicts ACGU and, for example, M6A. So where would this model even fit here? Where the idea is that when you have here your sequencing, here you have your Fast5 reads, and then instead of feeding a canonical model that can only predict 4 letters, you should feed a model that can predict 5 or even more letters. And then finally, you would then do the same processing afterwards. You would get your reads, you can map them, and so on and so forth. So this is a bit what we have been trying to do in the lab. And actually, here there has been a very collaborative effort by several members in the lab who have developed what we call the M6A base caller. And this is a bit how it really looks. So basically, the idea is that for each individual read, you get a prediction, and each position and nucleotide has a modification probability associated to it. And what you can see is that at several positions, individual reads actually get predicted as M6A modified, and that's why here you finally get the prediction of an M6A site at this position and not at others. And you can see that then you can even associate, which reads belong to which isoform, because this is a long read technology. So you can see, for example, if the here in the splicing patterns, if the intron is present or absent, if it correlates with the M6A modification, and so on and so forth. So we said, okay, we trained the model, but how does it work actually? Does it predict poorly, or does it do a reasonable job? So to benchmark it, we first turned into some in vitro synthetic molecules, which were either 0% modified or 100% modified here. Here you have the DRACA ACU, which would be the most representative one typically in human, as far as we know. And here what you can see is that basically the most of the reads are predicted as modified, whereas they are not predicted as modified in the modified control oligo. If we do the same in vivo molecules, what we see is that also the positions in the wild type are predicted as highly modified. And upon the knockout, you see a decrease in the modification stoichiometry. You don't see a complete loss. I'm happy to discuss why this is the case, but I think that the truth is that the knockout is not complete, because when we turn into in vitro synthetic molecules of the transcriptome, this signal is gone. So how does the global modification, because this could be just like kind of cherry-picked examples, how does the global landscape of M6A modification look using this M6A base caller? Well, what we see here, for example, is that, well, the replicates are actually highly replicable across independent runs. So you kind of get around a 10% modification stoichiometry. This is what we predict globally. And then you see that there's a massive decrease or shift in the knockout, which is not completely lost, but then there's like around whatever, 1-3% of modification stoichiometry as background signal. And something similar you can see in the case of yeast as well. So we said, okay, maybe let's see if we can get better or cleaner results with, our own data, because this was actually using publicly available data, not even our own data. So we decided to build a tamoxifen-inducible mouse CSC cell system. So here, basically, the idea is that we put some LOXP sites, and then afterwards we induce tamoxifen, which basically will remove the methyl-3. And here you can see how the methyl-3 protein is actually gone already at 6 days, but even more at 14 days. But basically at 6 days, the protein is already completely gone. And then to our surprise, well, we didn't see a huge shift in the modification frequency between our control and the knockout cells. And we said, like, okay, what is going on? Is our system not working? Is our model horrible? But actually we said, okay, let's also look into the 14-day treated cells. And then you do see the shift. And then this basically makes me think that what we basically propose is that a loss of protein in your Western blot, it does not yet mean that you have a complete loss of M6A in your system. So this means that we should be especially careful with all these inducible systems or siRNA systems and so on and so forth, because the loss of the protein seems to have a delay between the loss of protein and the loss of M6A in the RNA reads. So to further validate, finally, we said, okay, let's use even a methyl-3 inhibitor, the STORM inhibitor that actually was developed. And we used it at increasing concentrations. And here what you can see is that there is a clear decrease in the stoichiometry of modification at different sites here globally, with increase in the inhibitor concentration, really showing that the method is quantitative. Here you can see a shift going towards the left in the modification stoichiometry. The sites that are being predicted and lost are also meeting the classical location and motives that we would expect. So we were actually quite happy and think that the method really works in vivo and in vitro. So how does it compare to other things? Actually, like this has been a bit raised before. Of course, nothing is perfect, and we don't know really what is the truth of the truth, right? Especially in vivo. So you never know which is the one that is doing better the job. But basically, like what we do see is that the M6A base caller has a huge overlap with a bit of everything, actually. So huge overlap with GlorySeq, with MyClip, or with M6A-seq. These are different data sets from different people that have made publicly available their data. So thank you very much for that. And basically 91% of the sites that we predict are predicted by at least one of the other methods. And also there is some overlap to other nanopore methods, more to the M6A-net actually than to the XPOR, which are the ones that we use to benchmark the work. So one thing that we can now do, because we can predict modifications at individual reads, is to look into isoforms. And here I'm just showing an example of how you can see now that in individual, you can bin your reads by isoform, and then you can see that there's differences in modification stoichiometry. However, I have to say that in general, there are not like super huge differences across isoforms. So it doesn't seem that there's a huge regulation at the level of isoform specific modifications in our opinion, but we're still finishing the final analysis on this. And also you can do other things, like, for example, study what it is the interplay between poly-A tail length and M6A presence. This has been done indirectly previously by other works, but here really we can measure the tail length and the M6A presence in the same RNA molecule. And we see that there's a significant difference between, populations that have longer reads than those that have shorter reads in terms of like when you bin them based on whether they are M6A modified or not. Suggesting that there is an interplay between M6A modification and the adylation or poly-A tail length or stability, which is not really super news, because the role of M6A in stability in mRNAs has been already previously identified. So just briefly before to move on, I just also want to briefly mention the other challenge that we have been working on, which is to capture short RNAs using nanopore sequencing. So in general, when you look into nanopore sequencing, you can see that your reads typically will look like this, like long reads, reasonable coverage throughout, and then some errors, some can be systematic errors, others will be modifications and so on. And you get something like 1 to 2 million reads in a normal MinION. Now this is going to go up with the RNA 004. But then when you sequence things like tRNAs, we would typically get very, very low yields. And we were really struggling because we were interested in really understanding tRNA modifications in many different variety of contexts. And we wanted to sequence them. So we really put a lot of effort in trying to get tRNA sequencing to work. So after a lot of different optimizations, both in the wet lab and the dry lab levels, we finally were able to sequence tRNAs successfully. And here's just a bit of how it looks. So you can do it with the synthetic ones. There's still some background errors here, more than you would expect with long reads. But then when you go to biological tRNAs, you see a lot of errors, which are expected from tRNA modifications, right? So you pretty much get a modification, you get an error, and then, of course, because tRNAs are highly modified, you will have a lot of these errors through your sequence. So if we kind of do an aggregation of all your reads, we can then start looking into, like when we deplete a specific writer, how does that affect our tRNAome in a transcriptome-wide fashion, right? So for example, here, this is a structural alignment of different tRNAs. So this, for example, area gray here is going to be the variable loop of the tRNA. And then here, what you can see is that when we align all these tRNAs, and here would be like by the different positions here at position 55, you see that when we compare PUS4 knockout and the wild type patterns, you can see that there's a huge difference at this position, meaning that PUS4, what it's doing is something at position 55. We know it's pseudouridine, what it's putting. So basically we can say, okay, PUS4 is responsible for placing modification at position 55, which by mass spec, we already know that this is pseudouridine actually, right? And then you can do this with other writer enzymes. So here, for example, if we knock out PUS1, we see a different pattern, and it's not like present in all tRNAs like before, but more selective. And there's more than one position that seems to be affected. And the same happens here with PUS7 knockout, for example. And something that I'd like to briefly mention here is you see this kind of dirty pattern here after the pseudouridine 55. And this actually is, in our opinion, it's M1A, actually. So you can see that there's this position here, 55, and then in position 58, and a bit spread throughout several nucleotides, because not all modifications cause a single nucleotide base calling error, but sometimes it's spread around certain positions. When we bin the tRNAs based on whether we know they have M1A or they don't have M1A based on the previous literature, we can see that most of the tRNA that actually did not have M1A, don't have any error pattern here, and others do here. Really meaning that actually what we are seeing here is that the loss of pseudouridine at position 55 is accompanied by a loss of M1A, meaning that we can also start studying codependencies between different modifications with the same technique and in the same individual reads. And finally, just with this, just to thank the people who actually did the work. So this is really a lot of team effort. Pretty much everybody in the lab has been involved in all what I've been presenting today. And also to thank you all for your attention as well as the funding. And thank you. And I'm really happy to take any questions.
Let's see. I guess I can start with questions from the chat, and then I have a couple questions of my own. So Yuzu asked, does the M6A base color also detect non-DRACH M6A sites? Is there a difference between DRACH and non-DRACH efficiency detection?
Yeah, so the M6A base caller was trained to detect DRACH motifs specifically. So the thing is, you will train a model to basically detect whatever you train it with. So one can train the model in many different ways. The one that we chose to train will be preferentially detecting DRACH motifs, basically.
And then Yurin Kim had a question about how do you deal with the incomplete knockdown problem? And I was curious about that too. You were mentioning the metal 3 knockout cells, and I mean, I was surprised that 6 days in what the system you generated, it seemed like you had nearly complete loss of metal 3.
Yeah.
I don't
Yeah.
know if you validated the M6A levels like with mass spec or anything like that, but this seems to be an important issue, right? Because if you're using this as your benchmark, then this is an issue for all modifications that the field's going to have to deal with.
Yeah, so I mean, I actually wanted to share this because I think it's really important for everybody, and that, especially interesting and for people who are using these kind of models as well, because we were very shocked as well to see this result. We did the validation using mass spec, but then mass spec does show a much more significant decrease, and the reason is because what we see when we look into the 6 days is that those modified sites that are modified at high stoichiometry levels are the ones that are largely lost. But then most of the sites actually globally are modified at low stoichiometry levels, and these take much longer to kind of decrease. So let's say that if you decrease by whatever, let's say 70% your levels, well, that's, like if just a few sites that are highly modified can really account for that loss very quickly. But then like globally, most of your sites are still modified. And if you do things like ChIP-seq or RIP-seq or all these kind of, sorry, not ChIP-seq, but RIP-seq or any technology associated to antibody that will enrich for the M6A ones, you will still see the peaks because there's still a lot of reads that are modified at a stoichiometry similar to those that are under normal states, which, we are seeing it's something around 10%, right? So there is a shift in the curve. It's not that it's not present. And by mass spec, it looks like a huge shift because what we see is that the highly modified sites are preferentially lost. But those that are like, let's say at lower stoichiometries, which is the majority of M6A sites, actually are still present in the sample.
Well, and I guess it speaks to the function of M6A in those RNAs, right? Like we know that a major role for M6A is to promote degradation, but there have been plenty of examples in the literature of some M6As that can actually do the opposite and stabilize RNA. So that can obviously play a role too. And maybe even in that sense, you could use this as a way to kind of predict function for certain transcripts. Yeah,
I totally agree. So yeah, it seems that there's like a different behavior and maybe like, the role of highly modified sites is not the same as those that are modified on much lower stoichiometries.
Right. Okay, we'll ask one more question about your tRNA-seq data, even though we're going to be talking more about tRNA-seq tomorrow, but less so with nanopore. So I think it's good to address today. So Matthew Blango asks if you can comment on extending your nano tRNA-seq into new organisms or new systems.
Yeah, so this we didn't show in the work that was just published, but we have tried in other species and it works equally well. So we have tried mouse and human, and basically it's the same principle and it works equally well. You just need more material of input because the proportion of tRNA is going to be lower than in yeast, basically. Yeah. But apart from that, it's the same.
Great. Well, thank you again. And I think we'll have some more things to talk about for sure in the panel discussion that's going to follow here shortly.
Awesome. Thank you.
Thank you very much.
Thank you.
We have our last speaker for the speaker session before we have a brief break, and then our panel discussion is Dr. Logan Moroni from the Italian Institute of Technology, who's going to be telling us about NanoCompor and using that to look at RNA modifications.
So, good evening, good morning, good time of day it is for everyone tuning in. Thank you for the introduction and the chance to speak here at this meeting. I'm super excited to talk about some of our work here at IIT and the EBI. So as the three speakers before me have introduced fairly well, RNA modifications are chemical changes to canonical nucleotides, and they can occur internally or on the ends of RNAs, and they exist in pretty much all RNAs. And these modifications are involved in a lot of different processes for RNA. And one of my favorite examples is in the microRNA LET7, where an M7G is required to break a G quadruplex in order to process that microRNA. And without the M7G, the microRNA doesn't get processed and it can lead to a disease state. And so understanding where these modifications are and what they're doing for the cell is something that we all like to study. And so you've seen this now from Ava, but the strands are moved through the pore, one nucleotide at a time, regulated by this motor, moving the nucleotides through this pore. And it generates this electrical data. And I just want to point out that it's really important that the modifications cause an ionic current different compared to the canonical signal. And this is in principle what we're doing to detect the differences and how we find these RNA modifications. And so this is E. coli 16S RNA, where the writer for this M7G was knocked out, or the wild type where the M7G was present, and it creates this large difference here. But as Ave also pointed out, the difference can occur in adjacent positions as well. And so I'm going to talk a little bit about some software that Adrian Leger and Tommaso Leonardi wrote, and then I have inherited from them, called NanoCompor. And this uses a comparative framework to detect RNA modifications by comparing like a wild type sample to an IVT or an enzyme knockout or knockdown. And it doesn't rely on any kind of model training, so it makes it compatible with any kind of future ONT developments, such as the new RNA-004 chemistry that will be coming out in the relatively near future. And this gives you a fairly flexible experimental design when you're trying to compare these different samples to each other. And so just briefly how the clustering algorithm works. We have our two samples. So in here is just 5 reads as an example from a wild type sample, and then 5 reads from where we knock down the M6A writer, so metal 3 in humans. And so you get a different number of M6A sites divided between these two samples. And when you compare the ionic current features at a position that isn't modified in 2 dimensions, so we're comparing the signal intensity to the dwell time, all of the reads from these two samples will cluster together and we'll get one cluster. And so we'll call this as not modified. But when we compare the ionic currents for the position that is modified, these 5 reads will separate in one or both of these dimensions. And so we will find 2 clusters here. And then we perform a statistical test. So we do logistical regression of the original sample labels. And we look for an enrichment for one of the labels in one of the clusters. And with logistical regression, we can then get a p-value and then call that site as modified if we see an enrichment of one of the labels. And one thing I want to point out is the reference sample that you use with NanoCompor is really important for interpreting the results that you get from NanoCompor. So in the event where you do an enzyme knockdown or you manipulate one of the RNA modifying enzymes, so either a writer, eraser, or what have you, only the modification that is dependent on that enzyme will change, and the modifications that are not dependent on enzyme will stay the same. And so you won't get a signal difference between these two, and they'll be called as unmodified. Whereas if you do something like an in vitro transcribed RNA where all sites are canonical nucleotides, the modified positions at all of them will come up with signal differences. And so you can find the positions of all of these modifications. Importantly, though, the identity of these modifications can't be determined by NanoCompor. You have to use prior known information or some other source of information to identify them. And so this is an example where in 3 different sequencing experiments, we could detect a variety of different RNA modifications. So it's not specific to just one type. And so we have 3 100 nucleotide synthetic oligos that each have 3 RNA modifications, 3 M6As in the case of this oligo, an inosine, an M5C, and a pseudouridine, or an M62A, M1G, and a 2'-methyl A. And we compared these against a 4th oligo that had no modifications. And so we use that as a reference site, and we can find a peak called significant M6A site, modified site in all except for this one, which is 1 off. And so this shows that we can work with a variety of different modifications depending on the experimental setup. And in human cells, when you knock down the metal 3, you can find M6As. And so this is an example transcript of beta-actin. And this is a reverse orientation gene. So you have the 5' end here on the right and the 3' end on the left. And I'm overlaying the log transformation of the NanoCompor scores on top of the gene model annotation. And then these 3 larger peaks here, zooming in, which I'm calling site A, B, and C. And these peaks are centered in the canonical DRAC motif that metal-3 recognizes. And so we're fairly certain that these are indeed M6As and dependent on metal-3. And so because this is a long-read technology, we can start asking questions about the codependency of these sites on each other. And so when we look at the probability of modification for individual reads, which are in the columns here, for those 3 sites, in this B, C, and A based on the clustering, for the wild type on top or the knockdown on bottom, we can start looking like for this one, it's unmodified, modified, modified, or 3 positions not modified, or 3 positions likely modified. And in the knockdown, you're still getting some M6A modification, but it's definitely reduced. And so we then compared the observed frequency on every single read for all of the different combinations that we can expect in the brown bars here to what we'd expect if these were randomly distributed based on the counts for the reads that were found here in the dotted lines. And in this example, for beta-actin, it appears that the M6A sites are indeed randomly distributed, not dependent on each other, at least in this example. And then another example that I particularly like from this paper was looking at 7SK, where a specific adapter was used to target this molecule because it doesn't have a poly A tail, and then sequenced in the metal-3 knockdown experiment. And so what I'm showing here is a 2D structure of the 7SK, and highlighted in these boxes are where the RNA binding proteins interact with the 7SK molecules. And then here on the right is the same 2D structure, but now overlaying the nanocompor p-value for the log transformation. And what I find super interesting is that a lot of the M6A probability sites are at the same position where these RNA binding proteins interact with 7SK. And so it's possible that this modification is either used to promote or downregulate the binding of these proteins to 7SK. And it could be used by the cells as a switch to turn on or off binding. This wasn't explored in this particular study, and further experiments would be needed to test this theory, but I think it's interesting nonetheless. And so shifting gears a little bit, we've also used nanocompor with SARS-CoV-2 RNA. And so this is just kind of an overview of SARS-CoV-2. I think pretty much every genomicist has tried doing some analysis with this virus in the last few years. But one thing that's really interesting about the SARS-CoV-2 is how it's a single 30 kb RNA viral genome that then transcribes these subgenomic RNAs that are all subcopies of itself with a shared 5' end. And then these different subgenomic RNAs are used to make the different viral proteins. And it's been shown by others using a variety of different techniques that SARS-CoV-2 is highly modified. So in this experiment, using MuRIP to find modifications across the viral genome, and then looking at mass spectrometry data to look at the percentages of the different modifications identified in the viral genome. And what stood out to us is that pseudouridine is the most abundant modification in the genome by percentage. And so we wanted to explore that a little bit further. And this was an amazing resource produced by Kim et al. In 2020, where they made an in vitro transcribed reference sample from direct RNA nanopore data for the whole viral genome. And what they did is they layered these tiles of templates across the entire genome and then transcribed RNA from each of these templates, and then got relatively even coverage of the whole genome using this in vitro transcribe method. And so we used this as a reference sample and compared it to virus extracted from different cells that we cultured in the lab. And so we cultured in the CACO2, the CALU3, and the VERO-E6 cells. And this is a reference assembly of the SARS-CoV-2 subgenomic RNAs. And then each of these little points, all of these hashes, is a uridine-containing modification site by nanocompor mapped onto the subgenomic RNAs. And so we can get this isoform level information from nanocompor. And we focused on the uridine-containing sites because pseudouridine was the most prominent modification in the virus. Now, there are 14 known pseudouridine writers, And so instead of systematically knocking out or knocking down each of those 14 and trying to run Nanocompare again to see which sites are removed, we decided to instead look for which proteins are interacting with the virus. And so this is a technique called proximity labeling, where you have a target RNA that is flanked by these bbox stem loops. And then this BSU construct hybridizes with the stem loop, and when incubated with biotin, will label all of the proteins that are interacting with your RNA fragment in a very rapid manner. And then you can pull down using streptavidin, and then do LCMS-MS to figure out which proteins were present in your sample. And so using that technique to get our protein interaction index, and then comparing it also to an algorithm that a group at IIT came up with called CatRapid to predict RNA protein interactors, we got a list of several different protein hits. And this is just looking at the top 15%, so the 85th percentile of our hits. And this enzyme PUS7, or pseudouridine synthase 7, came up 3 times in our top 15%. And so pseudouridine synthase 7 is an enzyme that converts uridine to pseudouridine, and it targets the UGWAR motif. And so we use this information to further filter our uridine-containing sites to positions that were containing the UGWAR motif. And then we also filtered where the position had to occur in 2 out of 3 of our cell line culture. And so we could get a list of high confidence sites that were most likely pseudouridine. And so we found 54 positions where each of these lollipops on these different subgenomic RNAs represents one high confidence site. And one of the positions that interest us a lot is this pseudouridine at position 54 in the SARS-CoV-2 TRSL. And so here's a 2D structure of the first 80 nucleotides of the TRSL. And position 54 is right here at the end of this loop in stem loop 2. And so this is also one of our high confidence Nanocompare hits. And it's been shown that the non-structural protein 1 interacts with this region. Specifically, it's been shown to interact with stem loop 1. And we have some data on that pseudouridine in stem loop 2 changes the KD. But I don't really have too much time to show that right now. But so this protein does interact with this region. And we're interested to see if that pseudouridine, how much of an effect it has. And so in HEC293 cells, we expressed our fragment 1, which is the first 500 nucleotides of the SARS-CoV-2 genome. And so that includes this TRSL sequence. And then we also expressed the NSP1. And then we expressed 3 different constructs, either an empty flag tag, a flag tag pseudouridine 7 or pseudouridine synthase 7, and a flag tag PUS7 that is catalytically disrupted. And looking at the Western blot here, the protein expression for our 2 different pseudouridine synthase 7 constructs are about the same. And the NSP1 is roughly the same when normalized for our loading control. And so then we did a UV rip, which is where we cross-link the RNA to the proteins interacting with it. And then we used antibodies to pull down on the NSP1, broke the cross-link, and then made cDNA from all the RNA that was bound, and then used qPCR to quantify how much of the fragment 1 was bound to the different proteins. And so all 3 of the PUS7 controls against RPLP0 were about the same. And then the amount of RNA that we found with the empty flag versus the catalytically active PUS7, and then the catalytically disrupted PUS7 was reduced. And so it does look like that pseudouridine is important for NSP1 binding to this region, and that could control the translation of the viral proteins. And so this is just a really nice example of how nanopore sequencing and biochemistry assays can be used together to start learning more about the biology going on in SARS-CoV-2. And so with that, I'm going to conclude that I think with all 4 talks here in this session is fairly convincing that RNA modifications can be detected with direct RNA nanopore sequencing. The reference sample is really important for how to interpret the nanocompore significance sites. And then pseudouridine is an important modification for SARS-CoV-2 biology. And this interplay between using sequencing and direct RNA sequencing and modification detection in biochemical assays can support each other to learn biological phenomena. And this work would not be possible without many, many people all contributing to it. And so my lab at the EBI and then also in Italy at IIT have been fantastic to work with. And then I really want to thank Camilla and Roberto and Elsa, who have done most of the work on the SARS-CoV-2. And then Tommaso and Adrian, Tony, Luca, and Paolo, who did a lot of the work, or all of pretty much most of the work on the initial nanocompore development. And then my funding sources, which I would not be here without them. So thank you all for your attention, and I will take any questions.
That was fantastic, Logan. Thank you so much. So I think we have time for a few questions. And I actually have a question. So, you know, we can get into this, I think, somewhat in the panel discussion too, but, you know, we've heard, you know, already today from different speakers who have talked about their own methods as well as referenced others in the literature for analyzing, you know, nanopore data. What in your mind, you know, makes nanocompore stand out relative to the others?
So it's one of the few comparative samples that are out there. So that would be like XCOR, YanoComp, and nanocompore. And that gives you an advantage for looking at kind of modifications that are not common, I guess. So if you don't have a model built for it, like Ava's M6A base caller, or if there's a pseudouridine base caller that will be made. So one of those modifications that people aren't specifically making dedicated callers for, you can use one of these comparative models to find where those maybe less popular modifications would be that might still have just as much importance for the cell. And then a lot of the other methods rely on neural network training. And so as ONT keeps changing their chemistries, or their base color versions, which they put out a new version about every 6 months, nanocompore can keep pace with that because it's an unsupervised method.
Right. Yeah, to me, I mean, that seems like a big advantage. I'll be the first to tell you that we don't do any nanopore sequencing in my lab, but you know, we do enough other sequencing to know that these technologies are constantly changing and improving. And I think nanopore especially will be, since there's such an increased push from, you know, the academic community and from industry to start moving toward direct sequencing of RNAs more and more. So I think that to me is a major advantage of nanocompore.
Yeah, all of the tools have their niche. Yep. And so I think ultimately it's going to be using a combination of them to build confidence in finding the particular sites of interest.
Sure, I agree. And I think things like standards and benchmarks are going to be critical, and we can maybe save that discussion for the panel because I think that's really important to talk about. I think we have time for another question here, so I'll go to the Q&A. Yurin Kim asks, is it possible that nanocompore cannot detect M6As if your knockdown didn't work out well? And this is something that, you know, Ava kind of talked about a little bit too in her data and the importance of that.
Yeah. So with the synthetic oligos, Tommaso did a test where we can detect a difference of 10%, as long as, and it's a relationship between stoichiometry and coverage.
Sure.
The higher your coverage, the smaller the difference can be and still detect it. The lower the coverage, the bigger the difference needs to be to detect the site. And kind of around 100 reads in both samples is kind of that point where nanocompore becomes much more reliable at the smaller differences in stoichiometry. So in this particular case, it's more data is more accuracy.
Right. And I think that's the case too with, you know, non-direct RNA sequencing as well. It's almost always, you know, the more coverage, the better, although in some cases that's not the case. But yeah, especially when you're calling these substoichiometric modifications.
Mm-hmm.
Okay, great. Well, I think we have a break scheduled right now. So we will pick up at, let's see, 3:20 to have our panel discussion. And I think unfortunately, Dr. Goki is not going to be able to join us, but he is going to be answering, or perhaps he already has the questions in the chat, but he also said to make sure everyone knows, don't hesitate to reach out to him by email if you have other questions for him. So we will see everyone back here at 3:20.